Okay, so experiment 13 was acid-fast staining. And the purpose of this experiment was to learn how to distinguish acid-fast bacteria from the quote-unquote regular bacteria that aren't acid-fast. And so this is the last of the triad of differential stains from chapter 11 to 13. So there's one more to come. It's experiment 20. But that doesn't come till later. So what is an acid-fast cell? They are types of bacteria that don't lose stain once they are stained, and they are tricky to stain in and of themselves. And so the poster child of the acid-fast family is this Mycobacterium genus. They are aerobic, non-endosperming, non-motile rod-shaped bacteria that have these things called mycolic acids on their outside. And these mycolic acids, as you can see here, are hydroxylipids that are associated with the microbacterial cell wall. So, as you can see, they're very hydrophobic, which is an important characteristic of it. So they're just hydrocarbons if you've taken or are currently taking organic chemistry. And what this allows them to do is they're pathogenic. There are several strains of or not strains, but species of Mycobacterium that are pathogenic to humans and even other animals. And so they're very important to the whole microbial world, and so it's important that we go over them. They form these kind of cultures in this like cellular morphology of kind of this like crumpled. It almost looks like a crumpled up piece of paper. This is Mycobacterium smegmatis. As you can see, M smag is what they were going for. It's kind of art, I guess, in the plate, which is creative of these people. But yeah, and these crumpled up like colonies lend themselves to the fact that they have these hydrophobic mycolic acids on their outsides, and we'll get more into that later. So, because these are pathogenic, finding a way to efficiently diagnose them, to find them, is very crucial in diagnosing people who are infected with this stuff. So, what they find is that gentle heat helps in the staining, and so they found a way to gently heat up this stuff and then add stain to where you can stain the acid-fast cells. And so what you do is you use egg albumin, not water, which is important. We'll get into that later. And then your primary stain is carbyl fuchsin, which is a combination of phenol and fuchsin, which is a reddish pink dye. Phenol, if you remember, you might have gone over it in maybe organic chemistry or even microbiology class. It looks like a benzene ring with a alcohol group. If those words don't make sense to you, just ignore that. The next thing is the decolorizer. It's acid alcohol, which is very strong stuff. It's HCl, which is, as you know from general chemistry, a very strong acid. And then ethanol, which is a little bit weaker of an acid, but still slightly acidic. And then your counterstain is methylene blue. And so why do we use these specific things? So egg albumin, if you remember from back here, these are very hydrophobic things, and so water doesn't mix well with them. And so we have to use egg albumin, which is, I think, protein-based, in that it kind of acts as almost an emulsifier. An emulsifier is something that can combine hydrophobic and hydrophilic things. But what's important here is that it doesn't dissolve it, but it allows the mycobacterium to be suspended in the culture, and like water does to regular cells, just water can't work here, so we use egg albumin instead. And so this is the one way we combat this hydrophobic nature of the outside layer of Mycobacterium. Another thing that comes about in our studying of Mycobacterium as scientists is that it's hard to grow on artificial medium, because artificial medium is you can kind of conceptualized as hydrophilic, and so these hydrophobic outside layers of mycolic acids make it hard for nutrients in artificial medium to get through into the cell, and so they're very slow growing if they do grow, and there's even some mycobacterium that don't grow well on artificial medium. And so that just presents one problem. But the first step is egg albumin, to like suspend the bacteria, and then the carbofuscin is the main stain. So that's a reddish pink stain. It looks a lot like safranin, kind of, a little slightly darker, but it looks a lot like safranin. And then you use gentle heat and phenol. So here we come back to phenol. I don't know, when I took the lecture class, I was told a story about how the professor that she used to work for used to mouth pipette phenol and this nasty stuff. And so if you actually look up like phenol injuries, it's kind of gross if you do Google images. But I remember she used to tell us stories about how the professor used to like mouth pipette phenol, and he was apparently he was an older dude, and that's just how they did it. I don't know, but yeah, and so you want to use phenol because of its like reactivity. If you use a little bit of heat and a little bit of phenol, you can actually drive some of this stain into the cells, and so that's where this carbofuscin comes into play. Acid alcohol is the next step. It's very powerful decolorizer that doesn't work on acid-fast bacteria, and that's the key right here. So it works on non-acid-fast bacteria, but it doesn't decolorize mycobacterium like the acid-fast bacteria. So the acid-fast cells will remain red, indeed. And so this very powerful decolorizer, acid alcohol, decolorizes everything else, just not these acid-fast cells. And so we counterstain those non-acid-fast cells with methylene blue. And we don't use safranin. Typically, our go-to counterstain is safranin, right? We do that in the not only the gram stain, but also in the endospore stain. But here we can't use safranin because it looks so similar to carbofuscin that we need to use something else that has a different color. And so we use this methylene blue, which has this like bluish color, obviously, blue. And so it is a different color than the carbofuscin, so it allows us to distinguish the acid-fast red cells from the non-acid-fast blue cells. And so what we did was exactly that. We put the egg albumin on the slide, and then we put both the acid-fast bacteria, Mycobacterium smegmatis, and the non-acid-fast bacteria, E. coli. E. coli is kind of the how do I put this? E. coli is the bitch bacteria in all these tests because it's always used as the comparison because we know so much about it that we know what it does and doesn't do very easily, and it's very easy to grow, and it's really cheap, and it's just go-to bacteria for contrast in a lot of these experiments. So keep that in mind when you're studying. You might not have to memorize as much about E. coli as you think because it's always the not one. So it's not the acid-fast bacteria in this case. And so we put both of those on a slide with egg albumin, and we put a paper square over it with carbofuscin and boiling water. The paper square is to keep it moist, both in the endospore stain and in this one. And the boiling water is that gentle heat that we talked about. So we rinse with water and acid alcohol, and then we rinse with water, and then we counterstain it with methylene blue, and then we got something that looked like this, and then we see that this is our red acid-fast bacteria, and this is our blue non-acid-fast bacteria. In our case, it'd be E. coli. And so there's another picture in your lab manual of the acid-fast stain, just at the beginning of the chapter, page 59. And so the takeaway here is that we go through this whole process because mycobacteria tend to be generally dangerous things. You have things like tuberculosis and Hansen's disease that come about by mycobacterium species. And so it's hard to work with because it's hard to grow because of the hydrophilic. It's hard to grow in artificial media because of the hydrophilic outside, and then hydrophobic outside, and then it's hard to differential stain with gram stains, and then it's hard to take the stain out of with things even as powerful as the acid alcohol. And so we do this whole acid fast stain, and this allows us to diagnose things, people that are infected with maybe tuberculosis or some other mycobacterium, and then we kind of solve this problem and we are allowed to cure these people. So that's acid fast stain in a nutshell. Again, read your lab manuals, and then go over the specificities there.